Okay. All right. Good morning. I'm Vasant Honavar. Today we are going to talk about or introduce biomedical data sciences, in particular for biomedical audiences, right? And this is part of a series of multiple talks and presentations that is scheduled over the next several months. Okay, so let's get started. Right? So what is data and what is science? Right? Let's start with the very basics. So data is simply what you collect from measurements or observations. And science is the process of systematic studying of whatever phenomenon that you are interested in, whether it's biological, cognitive, social, or engineered systems. And using observation and experimentation, we generate data, right? So there is a whole scientific process that we utilize all the way from, starting with a hypothesis to collecting data, making predictions, um, building models from data, using those models to make additional predictions. And all of this is done in the context of some background knowledge. Right? So this data science as an enterprise began more than 200 years ago, even though it wasn't called that. It began when Tycho Brahe collected 20 years of extremely accurate astronomical measurements about positions of stars and planets. So this gave us the first set of big data. And Kepler, working for Brahe, fit this data in every way imaginable. Right? He was doing essentially what we now call machine learning, and he was doing it by hand. And then, so that led to the discovery of some regularities. And then it wasn't until Newton and Leibniz invented calculus that we actually had a language for talking about physical phenomena. And that led to the transformation of what used to be called natural philosophy into physics. So what used to be philosophy became science. Okay, this transformation is playing out in other fields now. So there are many fields which are at a stage comparable to what physics was 200 years ago. They're just at the beginning of collecting large data sets. There are no um, laws like physics. We don't understand, for example, in any precise way, how the different factors like genetics and environment and so on impact a health risk, for example. You don't have the equivalent of F equals MA for health status, but it's potentially possible when you have these large data sets. Right? So data science is a discipline that's concerned with collection and analysis of data to make sense of the world around us. So these data may be generated by humans, surveys, logs, etc., machines, CT scans, ICU data. Data may be in different formats, text, audio, images, video, and so on. And the analysis can be descriptive, predictive, or causal. So we will get into this in a little bit more detail shortly. So in some sense, data science is an intersection of computer science, which provides the algorithms for dealing with large data sets, math and statistics, which provide sort of the mathematical tools, and last but not the least, domain expertise. So for example, if you're practicing data science in the biomedical context, you need expertise around particular areas of medicine. Okay, so data science and artificial intelligence are closely intertwined when we talk about biomedical research. So artificial intelligence is a study of computational models of intelligence. It's about proposing, testing falsifiable hypotheses about intelligent behavior. It's about constructing artifacts that behave as if they're intelligent. It is about automating tasks that require intelligence. So for example, medical diagnosis. It is about augmenting and extending human abilities. For instance, an AI system working alongside a person, a physician, to improve, enhance the physician's performance. And it's about exploring the design space of intelligent systems. Right? The data is everywhere. So our instruments of observation have become extremely sophisticated. Data acquisition is increasingly automated, and there is exponential increase in the amount of data, the rate at which it's collected, the variety of data, the different sources. So for example, electronic health records data, physician notes with images, CT scans, the EEG signals, right? So you have many different data sources with their own semantics, their own meaning. that is brought to bear in analysis. And there's scientific literature. And so what is different in recent years is that data that was gathered for one purpose is being repurposed to answer questions beyond the setting in which the data was gathered. An example is, for example, electronic health records data, right? Initially, it was not meant to be used for research. It was meant as essentially a billing device. And now, uh, uh, lots of studies are leveraging that data to answer questions about health. Okay. So there's many different types of data, as I said, and biology is a classic example, which has been, I think the data revolution in biology began about 30 years ago, and it's in its sort of full force now. Some other fields are lagging a little behind, for example, humanities, and then healthcare is somewhere in the middle. Right? So in some sense, right, Rutherford said, all science is either stamp collecting or physics. It's a favorite quote of mine. Data revolution, this big data sets, dramatically increase the volume, velocity, and variety of these stamp collections. So data is like stamp collections. Okay? They're beautiful. And computing revolution enables us to sort, label, organize, and catalog and annotate these resulting stamp collections much more conveniently than and efficiently than before. The connectivity revolution, which is the ability to share data, interconnect data sets, interconnect databases, and so on, allows us to sort of link these stamp collections, link these massive data sets. But without data science methods, all we are left with are these beautiful stamp collections. So the journey from stamp collection to physics, the stamp collection to predictive modeling, from collecting data to understanding our world, whether it's biology or medicine, is impossible without data science methods and tools. So let's turn the clock back. This is 1970s, mycin. I don't know if anybody in the audience has heard of this system, but it was the very first AI system that was developed at Stanford to do essentially assist physicians with diagnosing blood infections, right? Bacterial infections of the blood. It was developed by Ted Shortliff at Stanford in the 1970s. And the way it was built was by very painstakingly interviewing physicians, expert physicians, and extracting what they knew about blood infections and encoding that into a collection of rules that the system then applied to diagnose blood infections. So this mycin informed itself about patients by asking questions about symptoms, history, lab test results, and so on. It was very interactive. The questions it asked was based on what it currently, whatever it thought was plausible hypothesis about the nature of the infection and the information it had. And it could explain its reasoning in English, in simple English, right? And there were studies that were done back in the 1970s that showed that mycin matched the performance of human specialists in blood infections and outperformed general practitioners. Now, you might say, if that was that good, why did not get to see broader deployment, right? Why did that not happen? There were several reasons, one of which is that building systems like that, because it involved working with human physicians and extracting what they knew, was extremely tedious and time-consuming process. And second, the world was not quite ready for AI systems being deployed in the clinical setting. Okay, so it's not long after the birth of AI 50 years ago that potential for AI to transform clinical care was recognized. Right? So this is a quote from a paper that Schwartz wrote in 1970 in the New England Journal of Medicine. Computers would be augmenting and in some cases largely replacing intellectual functions of the physician. That was 1970. But the designers of early AI systems attempted to distill the knowledge of experts into rules that the machine could use to perform the desired task, right? Whether it was diagnosing cancer or diagnosing blood infections and so on. However, these rule-based systems failed to live up to the hope that many had placed in AI. The reason was because coming up with these rule sets turned out to be too complicated and too expensive. Right? there was this almost 20, 30-year or period that was called the AI winter, that during which much of the research in this direction died off. But now AI is all the rage in healthcare. So what has changed, right? What is the difference? One key difference is that there has been tremendous advances in a subfield of AI called machine learning. There are many tasks like recognizing objects, driving a car, which we don't understand well enough to program machines, but we can get machines to learn from experience like humans and animals do. In the past, so this last decade has seen unprecedented advances in machine learning, which are essentially a marriage of statistical models and sophisticated algorithms. There's been tremendous advances in data acquisition. So the availability of large data sets. One example is the protein data bank, which fueled the creation of a system that for which the inventors got a Nobel Prize recently. And the convergence of these technologies is what has made applications of AI and data science feasible in biomedical sciences and healthcare. So there are many examples we can go through, but I won't spend a lot of time on this. So there is lung cancer diagnosis, there's cardiology, there is skin cancer diagnosis, lung cancer diagnosis, ophthalmology, and so on, right? And culminating recently in chemistry Nobel Prize, which went to the developers of AlphaFold. And AlphaFold is essentially a culmination of the creation of Protein Data Bank, an extremely large database of all experimentally determined structures, and a deep learning or machine learning algorithm that was trained on this large database to learn the regularities so it could predict protein structures, right? This protein structure prediction has been an unsolved problem for many years. We still don't understand, for example, the detailed physics of protein folding, but structure prediction is more or less a solved problem, thanks in large part to the availability of data and the advances in machine learning. Okay. So the potential for application is everywhere in healthcare, right? All the way from biomarker discovery, drug discovery, understanding the relationship between health and environment, understanding and addressing health disparities. The list is long, and the types of data are vast and extremely rich. And the current state of AI in healthcare, if we were to summarize it, is that it's not that AI is going to replace physicians anytime soon. And most people would agree that that's not necessarily a desirable thing. However, to quote a colleague, if you ask the question, will physicians be replaced by AI? The answer is very likely not, for many, many reasons. But physicians that don't understand AI may very well be replaced by physicians that do. So this is an important tool to understand. So why are we holding this series, this seminar series? All sciences, including biomedical sciences, are benefiting tremendously from advances in data science. All aspects of human endeavor, including biomedical sciences and healthcare, are being transformed by data and AI. All biomedical scientists and healthcare practitioners need to understand how to leverage data and AI, both to accelerate discoveries and to improve what they do, improve healthcare. So I already mentioned this, AI and data science offer tremendous potential for revolutionizing discovery in biomedical and health sciences, advancing all aspects of healthcare. But these advances require rigorous research. So it is not just a question of taking some off-the-shelf tool that was marketed by some commercial enterprise and blindly using it in hoping that it would work on your problem. So every application of data science or AI in healthcare is a research project, and it's something that you have to rigorously design and evaluate. So these studies, for example, a data science study starts with a planning phase. We decide what to measure, develop a study design. Then you go on and collect data. You do data quality checks, and eventually you analyze data at some level, maybe summarize the data, visualize the data, and so on. Eventually, you might build some predictive model, you interpret the results, and if it works well enough, right, you may go through this iteration several times, altering your analysis procedure and altering your data collection, the variables that you use, and so on. And if it works well, then you go ahead and maybe deploy it and do a clinical trial to demonstrate its efficacy, and then you use it, right? So this is sort of the process. It's not just some company coming to us and selling us a pre-made solution, which is a black box, which has never been validated, right? And this needs to be done for every problem, every new problem, every new question that you have. So the origins of data can differ, and data can be coming from a design study, an observational study, or an experimental study. There are also data that are in the wild, in the sense that you had no control on gathering that data. It just is there. It was gathered for whatever purpose. And you don't completely understand the story behind that data, why it looks the way it does. Examples are documents on the internet, electronic health records of patients, interactions on Facebook. These are data in the wild. And the data in the wild are far more challenging to work with than data from a design study, right? But you don't always have the luxury of collecting data from a design study. So you have to use the data that you have. And so we have to deal with data in the wild as well. And formulating questions, answering them, and interpreting the answers requires creativity, skill, and rigor. So doing data science is rarely simply a matter of blindly applying a tool. Okay. So when it comes to research or in general life, right, there is this famous quote by Feynman, the physicist. Right? The first principle is that you must not fool yourself, and you're the easiest person to fool. So not fooling yourself when you work with data requires data of adequate quality and quantity, understanding the story behind the data. So for example, if the data was limited to a certain subpopulation and you ignore that fact, then you're going to maybe apply the findings beyond its where it's applicable. Data science and AI competence is needed, domain knowledge and expertise in the relevant areas of medicine, and last but not the least, scientific rigor, rigorous analysis, quality control, reproducibility of the study, validation in many of these steps. Right? So that's the goal of this series, is to cover these aspects. So today we start by basically, the anatomy of a data science project, right? So this is a schematic that I took from a paper by Leek and Peng that appeared in Science in 2015. And it nicely summarizes what a data science or biomedical data science study, the flow of it looks like. Right? So there is, it's like a flow chart. So there are studies that simply are not that don't involve data analysis at all, right? So we exclude those. We are interested in data analysis. And in particular, there are different types of studies, some that just summarize the data, descriptive analysis, some that just are exploring some idea, right? For example, you might look at a small sample, you might look for some association between one variable and another, but you don't attempt to sort of make general claims beyond that sample. That could be considered an exploratory study. But if you want to quantify whether your discoveries are likely to hold in a new sample beyond the one that you looked at, then you are in the realm of statistics. You're trying to figure out how changing the average of one measurement affects another. So if it's purely the answer is no to this, right? So you're not interested in a causal mechanism, but just trying to predict the measurements for individuals. For example, you're trying to look at a patient's health record and predict, let's say, the risk of cancer, then it could be a predictive study or it could be an inferential kind of study. And then if you're interested in a causal effect, how changing some aspect of one's behavior impacts risk of some particular disease, then you are in the realm of either causal studies, which is purely with data, or mechanistic studies, which might involve control manipulation of the system that you're studying. So this is the broad scope of data science. The mechanistic studies are not something that we are going to necessarily look at in this seminar series, but everything else we will touch upon in some level of detail in subsequent seminars. Okay, so let's dive in a bit, right? So any study, any research project, any data science project begins with a question. So this is the most important task, right? If you don't have a good question, nothing matters, right? You can't decide what data to use. You can't decide what methods to look at. You have nothing to start with. So questions come from people that understand the domain. Right? Questions come from not from necessarily a data scientist, but from a physician or a biomedical scientist that has a pressing need to answer a particular question, right? So, and the question can be descriptive or exploratory, the many different types of questions. So here are some examples. So suppose you want to ask the question, what is the prevalence of lung cancer in Asian women, right? So this is a descriptive question. You collect some data, in your health system, let's say, and then you ask, this question. I mean, it's something that you can calculate from the data, and you describe your data, right? You may describe the characteristics of the women that have a high prevalence for lung cancer versus the not, and so on. You might have an exploratory study, which is basically, you're looking for evidence of some association. So maybe you have a sample of 1,000 Asian women, and they have, their smoking history, and you're trying to figure out whether there's some, you're interested in exploring whether there's an association between smoking history and lung cancer. Now, you may want to go beyond that. You may want to really predict the risk of lung cancer based on everything that you know about an individual. So somebody's smoking history, their genetics, many other lifestyle factors for that individual, and you don't exactly know how those different variables interact to make those predictions, but you believe that that prediction can be made, and you try to build a prediction model. Right? A prediction model is simply one that exploits correlations between variables to make predictions. It is not a causal model. Just because some variable is predictive of lung cancer doesn't mean that that variable is the cause of lung cancer. There are hundreds of papers where people go from essentially a statistical analysis of associations or a machine learning type of analysis that leads to a predictive model and jump to causal interpretation, and they are on really shaky grounds, right? And there are many, many examples of that in the literature. Or you might have an inferential type of question where does lung cancer risk change with lifestyle and socio-demographic variables in the US population? Right? So this, you have, all you can study is a single sample, but you're trying to extend your analysis. You're going to make some claims about the larger population from which the sample is drawn. And this usually relies on statistical methods, right? You have to look at whether the association is statistically significant, whether whether you can actually make claims about the general population based on the sample that you have studied. And then there are causal types of questions. So if you change some particular variable, so for example, if you switch your diet from high carbohydrate to a low carbohydrate diet, can you impact the risk of diabetes, right? So this is a causal question, and it can be studied in two different ways. The easiest way to study this would be a randomized trial where you take a population, split them into two groups that are statistically identical. One of them receives a low carbohydrate diet, the other receives a high carbohydrate diet, and you can look at the difference in the diabetes rates in the two subgroups, and you compare them and you get an effect. However, these randomized controlled trials are very expensive, and they may not be, in some cases, they may not even be feasible. So for example, you're interested in the causal effect of smoking on cancer. You can't obviously ask people to smoke just because you want to run a controlled trial. So you have to look at methods for getting at these causal effects from observational data. That's an area of data science where there's been tremendous progress in the last 20 years. And there are conditions under which, settings under which you can, in fact, tease out causal information from observational studies. And then the last part is the mechanistic types of studies. So where you actually want to understand the biological pathways and so on. And this involves more detailed experiments that kind of go beyond and require a much more in-depth understanding of the system that you're studying in order to be able to draw conclusions. So the anatomy of a data science project, you start with questions, right? Identify suitable data sets if available. And so this is like, if you want to answer the question of, risk factors for cardiovascular disease, you might be able to do that using historical electronic health records data. It's flawed, it's biased, but it is a useful set of data. Right. Or if an adequate data set is not available, you may want to go and gather your own data. And if you're going to gather your own data, then you better talk to somebody that understands study design, right? So you want to gather data of the type, and you want to gather data in ways that would lend themselves to analysis to answer the questions that you started out with. And then you proceed to conduct your analysis. And there is this dictum about not fooling yourself, which means really critically examining all your findings, and eventually you report your findings. And at all of these steps, there is questions of ethics, data ethics, data bias, whether your analysis is reproducible, and so on. So the data science comes in many flavors, as we mentioned. There's descriptive data science. So an example of the outcome of a descriptive study is, what is the distribution of cancer patients by race? How is gender correlated with incidence of lung cancer? Lots of these studies are done by many people, and there are hundreds of papers getting published just by looking at a data set that people didn't have access to before and doing some fairly straightforward analysis and understanding something about that population. Right? Then there is inferential data science, where you are interested in generalizing the findings beyond the sample that you have to the population at large. And this is where statistics comes in, and 100 years of statistics comes in here, and you want to talk to a statistician to really do this step rigorously. There is predictive data science, where the question is, can we predict lung cancer risk based on genetic factors, environmental exposure, lifestyle factors, and so on. And if you can predict, what variables are predictive, right? So this is where there are classic statistical methods from decades ago, but as the data has become more complex, you go from a dozen variables to tens of thousands of variables. Data sets that are instead of hundreds of samples, you have millions of samples. You change the types of methods that you use. They need to scale to large data sets. They need to take advantage. They need to deal with high dimensionality and so on. And so this is where the modern machine learning methods come into play, and applying them well also takes a great deal of skill. And then the last but not the least is the causal data science, which is teasing out causal relationships, which is relatively straightforward in the case of randomized controlled trials, but not so straightforward in the case of observational data. And that's where, again, modern methods in causal effect estimation, causal inference, causal modeling come into play. So in the case of descriptive data science, the goal is to characterize a data set, a sample from a population, right? And the primary tools are basic statistics, visualization, and so on. Inferential data science, you are interested in estimating parameters of distributions from samples. So you have a sample, but you really want to understand the population at large. And when you do that, you're making estimates, coming up with estimates. You're quantifying uncertainty of estimates, confidence intervals, and so on. Right? So this is the domain of much of biostatistics. And then there's predictive data science, where machine learning methods are used for classification, regression, and many other problems. There's a virtual zoo of methods. So they have their strengths and weaknesses. There is no single method that's going to be the magic bullet for every data science predictive modeling problem. So this is where a deep understanding of the methods, their strengths, and the weaknesses comes into play. And the choice of performance metrics and evaluation is incredibly important as well. So, if you just look at, for example, accuracy of prediction um, in a case where only a tiny fraction of the population that you're looking at has a particular outcome. So, for example, predicting cancer diagnosis, where 2% of the people have cancer, and and if you have a model that is 98% accurate, but gets the diagnosis for the 2% of the individuals that actually have cancer wrong, then it's not a useful model, right? So there is a great deal of attention has to be paid to how we are going to evaluate the models, um, quantifying the uncertainty around the predictions. And so this rigorous evaluation is incredibly important. And then there is many practical considerations, uh, missing data. So for example, when you look at electronic health records data, there are tens of thousands of variables. Uh, Majority of them are missing from any given visit, right? So only a handful of them are recorded. But the missing variables are not often missing at random. There is usually a mechanism behind missingness. So for example, if a COVID test result is not present, it probably means, the physician didn't think a COVID test was warranted for that patient at that time, right? So there is that. There is extreme high dimensionality, longitudinal nature of this data. So there's many challenges that are currently, even as of now, being addressed, and these are not completely solved problems from a methodological perspective. So causal data science deals with this notion that association is not causation. And if you can do a randomized controlled trial, that's great. But when that's not possible, we have to figure out how to, under what conditions you can actually get at a causal effect from observations alone. And there are methods for doing that under certain assumptions about the data generating process. And there are even techniques now for taking a study that was where a causal effect was found. Let's say a study with one population where a randomized experiment was run, and you found a causal effect, and you want to understand whether that same causal effect with some tweaks holds or not in a different population where you're not able to run a randomized control trial. This is the so-called causal transportability problem. So these are all areas where there's been tremendous progress in the last 20 years, and taking advantage of that in the studies that we design here could be incredibly beneficial. So I will conclude by just briefly talking about the plans for this series. So there'll be modules on various aspects of biomedical data sciences and AI. So immediately after this presentation, there'll be a presentation about study design. And then we have modules on various aspects of data science that I just went over. And eventually, we hope to have more specialized topics as well that are of interest to parts of the community, like whether it's image analysis, biomedical natural language processing, network analysis, different types of omics data, data integration, and so on. So with that, I conclude, and I'm happy to take questions.
Dr. Honavar, we would actually take questions once Alan finishes his presentation so that we don't need to share those on YouTube. So
Okay.
I'll ask everybody to hold on to their questions. We'll have time, 10 to 15 minutes, once Alan is done with his presentation. Thank you.
Sounds good.
Everybody hear me okay?
Yes.
Just got to find the,
Presentation mode.
yeah,
Yeah, all good.
good. Hello, everybody. My name's Alan Kunselman. I'm a biostatistician in the Division of Biostatistics and Bioinformatics and the Department of Public Health Sciences. And I'm here today to present a brief presentation on study design. And this is part of a series of lectures that the CTSI Biostatistics, Epidemiology, and Research Design Corps, in collaboration with Informatics, is planning to present over a period of time for a framework for initiating new research. So, welcome. So the outline of this brief talk today, I'm going to talk about types of non-experimental and experimental study designs, strengths and limitations of these study designs, the best study design for the goal that the researcher has in mind, and if there's time, I'll talk about some clinical trial study design considerations. So I just want to start off with a couple of quotes. Pocock mentioned in a paper of his that a clinical trial is any form of planned experiment which involves patients and is designed to elucidate the most appropriate treatment of future patients with a given medical condition. Followed up with Simon, this is about trials. The guiding principle of a clinical trial is to ask an important question and get a reliable answer. So in short, what we have is study design is a scientific method that a researcher follows to assess the association between an exposure and an outcome. And without having a proper study design, the results that you get will be questionable by reviewers and others. So you want to put some thought in what study is the best design for the question I have at hand. So the two main branches of study designs I want to talk about today in this brief presentation are non-experimental or observational studies and experimental studies. Non-experimental studies, the researcher observes both the exposure and the outcome. It's already happened or will happen. Experimental studies assign the exposures and observe the outcomes. So just to reiterate, a non-experimental study, you're observing both the exposure and the outcome. And an experimental design, you're assigning exposure. For example, in a clinical trial, you're randomizing patients to drug A or drug B, but you observe the outcome that they obtain. So in each of these branches, what types of studies are there? And there are more than I have listed here. This is just an overview. So for non-experimental, you can have case reports, case series, cross-sectional study, a case control study, or a cohort study. And cohort studies can be either prospective or retrospective, and I'll go to that a little bit further. And experimental design, the main one you're going to hear about is going to be a randomized clinical trial. And within that branch, there are a lot of different types of designs that we don't have time to go into today, but I'll just list a few. We can have a parallel design, a crossover design, a factorial design. And I should have mentioned, same with the non-experimental design, there are branches within those. So you may have heard the term a nested case control design. What that is, it's a combination of a case control study with a cohort study. So there are flavors and variants of each of these designs. So first I want to talk about non-experimental study designs where they are descriptive studies. And the main objective of these descriptive studies is the research to describe the main features of the population. And these are case reports. So what is a case report? It documents unusual medical occurrences that may represent the first clue in the identification of a new disease or an adverse event of the exposures based on one patient. So all case reports are on one patient or participant or individual. And I am coming from a medical field, so you may hear me use the terms patient or participant or subject a lot, but extrapolate that to whatever field you have. Study design is immutable. It's whatever the experimental unit is. If you're doing animal research, your experimental unit may be in a rat. If you're doing agricultural research, your experimental unit may be an acre of a field. The further on the case reports, though, it's only one patient. So there's no control for the exposure assignment. There's no control for the endpoint ascertainment, and there's no control for confounders. Further, there's no control group to compare this one patient to, and it's susceptible to large selection bias. Another descriptive type design is a case series. And all a case series is a collection of case reports. So it carries more weight than a single case report because it's based on more subjects, but again, susceptible to large selection bias. Another type of descriptive non-experimental study is a cross-sectional study. And a cross-sectional study is a study where the data is collected at one point in time or a relatively short period of time, and it reflects information on the prevalence of the outcomes. The big thing here is it assesses exposure and outcomes simultaneously in a defined population. So for example, if you wanted to say, I want to know everybody that came to my clinic in the month of November, and I want to know the outcome is, does this person have thyroid cancer? Yes or no? And the exposure could be, is the person overweight? Yes or no? So you're collecting that short period of time, 1 month, and you're just trying to ascertain some outcome and some exposure. So we're still in the non-experimental, like observational type studies, but rather than descriptive, we'll move on to analytical type studies. And the main objective with the analytical studies is you want a specific association is trying to be assessed. And two of the main designs here are case control studies, where the study participants are selected based on their outcome status. So you have a group, for example, you have a group having the disease of interest and another group who do not have that disease of interest. And the information is on past exposure is collected and compared between the two groups. As opposed to a cohort study, where a cohort study designs where participants are selected based on their exposure status. So you have a group having exposure of interest and another group who does not have that exposure of interest, and you follow them over time. So for a prospective cohort, participants are followed over time to observe the occurrence of an outcome in each of the groups. And a retrospective cohort, it's a similar idea, except the data has already been collected. So to put this a little bit clearer, so a prospective cohort, you could say, what's my exposure? Is this person a current smoker, yes or no? That's the exposure. And you follow them over a period of time, say 5 years, and see, do they develop lung cancer? Whoops, I'm sorry. A retrospective cohort would kind of be the same idea, is I have a database already. The data is already collected. I have both the exposure and the outcome, but I select the subjects based on their exposure. Did they have, in the year 2000, were they a current smoker? Yes, no. And then I look at that retrospective database and see, did they have the outcome of lung cancer? So you can have, that's the retrospective cohort. Case control, just think of the opposite. The outcome for a case control study is always going to be, are they a case or a control? So you're trying to see which exposures are associated with the outcome. Cohort studies are, start with the exposure and see what outcomes occur. And just to give you an idea, there are lots of ways to get information for observational studies, but here's just some databases. In the health field, you can look at the electronic health record, EHR. So that's detailed patient information at healthcare providers. Claims data. So insurance claim data providing diagnosis, treatments, and outcomes. And this is an area where would really help with informatics. This is big data. There are claims data databases have millions of records. So they're very large, unwieldy databases. Other data, national surveys, for example, the NHANES data, if you're familiar with that. Disease registries, like cancer registries. So all these are sources that you can look at for retrospective or observational studies. So if we move on to experimental studies, what's the experimental study? So the researcher assigns different exposures to participants based on multiple criteria, and it's usually a randomized trial, and you observe which outcomes occur. So here the researcher is actively carrying out an experiment on participants. And the main thing for experimental study is a randomized clinical trial. And that's the primary experimental study. So now that we've talked about the different types of these two studies, let's talk about some strengths and weaknesses. So what are some strengths of a cross-sectional study? Well, they're relatively easy to conduct because it involves identifying a group of subjects, collecting information on them, and analyzing the data. It's a study that can be done over a short period of time, so there's no follow-up needed on any participants. Because the data is already in a short period of time and you're not doing any experimental, again, you're not assigning treatments or anything, there's typically no ethical issues to worry about. It's data on all variables. You can look exposure, outcome, confounders, etc. They're only collected one point in time, so it's very focused. Prevalence may be assessed on all the factors being studied. Multiple exposures and outcomes can be studied. So you can have different types of cancers and different types of exposures and see what their relationships, if any, are. Prevalence of diseases or other health-related characteristics may be used to assess the burden of the disease, a specified population. And this helps plan and allocate health resources. And cross-sectional studies are very good for providing descriptive results for generating hypotheses for future studies. That all sounds good, but there's always limitations to things. So what can a cross-sectional study do? We can't make any causal inference. And this is mainly because the time sequence of exposure and outcome is not implicated in the design. So it's difficult to know which came first. Did the exposure happen before the outcome, or did the outcome happen before the exposure? So you're just going in at this one point in time and pulling data. They're not suitable for rare diseases. Back to my example of going in and see all patients in a month. If it's a rare disease, you go in there, it's possible you have nobody that has that disease in that short period of time or that one pool. So you don't want to use a cross-sectional study if your disease you're researching is rare. You can't estimate incidence, at least not easily. Again, because it's not a function of time normally. It's just at one point in time, you're looking at prevalence, not incidence. They're susceptible to a selection bias because of sampling and low response. If you go in and hit a survey, a cross-sectional, I'm going to send a survey out, you may get very low response rates. They're susceptible to information bias, and there are all kinds of information bias, like recall bias, asking people to remember things that they happened a year or two ago, non-response bias, that happens a lot with surveys, or errors in measurement that were collected when you're getting it. And they're also susceptible to confounding bias due to inability to collect information on all potential confounders. You may not have the resources or know which all confounders may be, so you may be missing some that are really important. So onto the case control studies. What are some strengths of a case control study? Again, they're relatively fast because they can be conducted with no follow-up, because both the exposure and the outcome have already occurred. Again, these are usually case control studies, or almost always retrospective studies. They're typically low financial cost, as all the data is already collected and or in a relatively short period of time, no follow-up needed. Again, as the data has already been collected, you're not performing an experiment or assigning any treatments. You don't have to worry about ethical issues. They're appropriate for studying rare diseases because the participants are selected based on the outcome status. So if your goal is, I want to look at a rare disease, a case control design is very strong, because you go in and say, boy, I really need 50 cases of this rare disease. At least with that, here's this big database, I can find these 50 cases, versus what we just talked about, cross-sectional. One point in time, you're probably not going to find these 50 cases. It may take you 20 years to find 50 cases in a very, very rare disease. They're appropriate for study diseases looking at outbreaks. Here's a really simple example would be, oh, here's a group of people that went to a restaurant and some of them got sick. So my cases are those that got sick at this restaurant. My controls will be those that did not get sick at the restaurant. So what are the exposures? Okay, if I'm familiar with the movie Airplane, did they have the fish or did they have the steak? So you're trying to figure out what were they served that gave them sickness. So it's good for outbreaks. And you can extrapolate that example to something much more serious and important than my movie Airplane example. And they're appropriate for assessing the association between multiple risk factors and one outcome. So again, as I kind of stressed a little bit, a case control study, the outcome is always, is it a case or a control? So you can look at multiple risk factors. Is weight associated with my outcome? Is gender associated with my outcome? Is whether the person is hypertensive, yes or no, is that associated with the outcome? So you can look at multiple exposures and see if they're associated with this outcome of interest. So what are some limitations of a case control study? Well, again, they're prone to selection bias if the cases and controls are not representative of the underlying population. So story takeaway here is, especially with the controls, you have to select your controls carefully. And you may have heard like matching and things like that, which I don't know if we have time to go into today. Case control studies are also prone to information bias because the information collected has happened sometimes long in the past. You may get recall bias and errors in measurement again. And the errors in measurement usually occur because the data has already been collected. You can't go back 10 years ago and ask the nurse that entered the data, oh, this number doesn't look right. Did you enter it wrong? So that's where we can get errors in measurement. Confounding may be an issue, as it may not be feasible to collect all the information on a wide range of potentially confounding variables. They're not appropriate for studying rare exposures, as you likely won't have enough subjects that will be exposed. So we talked about before, the case control studies are not good for rare exposures, whereas, go back up here, cross-sectional is good for looking at rare diseases. I'm sorry, it's not good for looking at rare diseases, sorry. Right. And you can't make causal inference with case control studies. So let's look at cohort studies. What are some strengths of cohort studies? Well, they're less costly and easier than experimental studies, but they're more expensive. They're kind of an in-between. Case control studies and cross-sectional studies are usually cheaper than cohort studies, but cohort studies are usually much cheaper than experimental randomized controlled trials. Cohort studies directly measure disease incidence due to follow-up. So if incidence is your goal, cohort studies is a good design. Temporal relationships can be established between the exposure and the outcome, so you can build evidence for causality. They're appropriate for assessing the association between one exposure and multiple outcomes. So again, you're selecting your cohort study selected on the exposure. So you have this one exposure, and it could be, are you a current smoker? But you can look at that one exposure with, again, multiple outcomes when they occur. Did these people that smoked or not smoked, did they develop lung cancer? Did they develop thyroid cancer? Did they develop emphysema? Are you still there? I lost my slide. Did they develop any other type of disease? They're appropriate for studying rare exposures. So again, just like the case control study, you can go in and find you're good for rare outcomes. Cohort studies are really good for rare exposure. And they're fast to complete if it's a retrospective cohort, because the data's already been collected. Some cohort limitations. Again, they are expensive, but they're not as expensive as a experimental study. And they take a long period of time, especially for a prospective cohort. Depending on what you're trying to research, you're following these patients sometimes 6 months, sometimes a year, sometimes 5 years. Depends on the goal of the study. You could have imbalances in subject characteristics could exist, thus introducing bias. They're affected by confounding because it may not be feasible to collect information on all potential confounding variables. They're not appropriate for studying rare outcomes. Again, you start with what the exposure is. And if it's a rare outcome, it may never occur in these subjects. So cohorts, not a good use of resources for a rare outcome. And loss to follow-up, especially when the duration of follow-up is very long, may introduce some selection bias. So on the experimental studies, what about randomized controlled trials? Well, treatments are compared directly to establish superiority. You can make causal inference. It's the strongest empirical evidence of a treatment efficacy. Randomization will minimize allocation bias and selection bias. Blinding mechanisms will minimize performance bias or observer bias. Double blinding minimizes assessment bias. Prospective design will minimize recall bias and selection bias because you're collecting the data in real time. Randomization will minimize confounding due to the unequal distribution of prognostic factors. Randomization makes groups comparable according to both known and unknown factors. You can do a stratified randomization, which makes groups comparable within known confounding factors. And they're basically considered the gold standard for publication, and especially for causality studies. But what are some limitations of RCTs? They're expensive. They can be very expensive. They can be very hard to recruit patients for. Many people don't want to be a guinea pig. A lot of studies will randomize people to an active treatment and a placebo. And a lot of people don't want to be randomized, but hate the chance to be randomized to a placebo group. Recruiting participants may take a very long time. There's much greater responsibility in documentation. So if anybody in this group has had an NIH grant, there's a lot of documents and recording adverse events and a lot of responsibility of your sponsor with randomized clinical trials. Typically require multiple visits over time. A long trial may result in a loss of relevance as the clinical practice may have moved on by the trial is published. So that you may start out today and the trial takes a long time. It's a 10-year study. By the time you're wrapped it up, a new method may have come out and it's obsolete, what you started with. Randomization requires clinical equipoise. So this is an ethical issue. In some cases, being my example of placebo, is it ethical to randomize a person to placebo, especially if they have a very debilitating disease? Participants may drop out. It could be an issue, especially for long trials, and compliance to the protocol could be an issue. So just briefly, of these trials I've talked about, what's the best study design for the goal the researcher has? Well, if the goal is to estimate the prevalence of a disease or prevalence of anything, cross-sectional study is the go-to study. Sometimes a cohort study, but cross-sectional. If the goal is to estimate the incidence of a disease, because you need time to do incidence, a cohort study is the best study to use. And sometimes you could use an RCT, so it's a function of what's a trade-off, resources and funding. If you're attempting to show causality, a randomized controlled trial is the go-to trial to do. And the last two, generalizability and feasibility. All of these can actually address these as long as they're carefully planned. For example, I have feasibility cross-sectional case control is probably the best. But if your goal in the feasibility is, can I recruit patients? Then you probably want to do a pilot feasibility randomized controlled trial to see how well can I recruit people? And the only way to do that is using an RCT. So on the topic of clinical trials, what are some considerations if you're actually doing a randomized controlled trial? Well, you want to control for the outside influences through appropriate comparisons. So again, you want to compare experimental treatment or placebo or a standard therapy to control for like a placebo effect. What are some types of controls? Well, they're historical controls or non-concurrent controls. And this is really frowned upon. So it's basically, there's different times, investigators, protocols, diagnosis. It's where you're doing an experiment on a group of people, but the controls you're using are data from the past. For example, I'm pulling this data from a paper that was published 5 years ago. It's going to be my control group because I can only afford to do my study on, I can't afford to do both two groups. I can only afford to do one group. I'm only bringing this type of control up because it has been done, but again, this is typically frowned upon. What you typically want to do is independent or concurrent controls. So this is like a parallel design. If you're doing a randomized study, you're randomizing people to the exposure or the experiment and another group to control, which could be a placebo or an active therapy. It could be self-controls. So again, you can have the same people get the control, and then over a period of time, they get the exposure. So the time effect can be confounded with the treatment. So this is like a pre-post design. Another design is a crossover design. So what you're including are self-controls and concurrent controls in an efficient design. So it requires a washout period. So in this example, let's think about a drug. We're going to randomize people to an exposure, an active drug, and a control like a placebo. They may be on that drug for 6 weeks. After 6 weeks, you wash them out. And this looks like, if you're looking at a drug trial, what's the half-life of the drug? So you want a washout period that all the effects of that drug are out of the system. And that might be, say, 2 weeks. Then after 2 weeks, the people cross over. So the people that initially started out with the active drug cross over into now they're going to get placebo. Those that start out and say the control placebo will cross over and now they're going to get experimental drug. So this way, every patient actually gets the experimental drug. So this is a good design that helps get rid of an ethical issue of not treating everybody. Another type of design is a factorial design. So here, a trial with participants are randomized to 2 or more treatments. So here, if we think of, we're going to randomize people to either active drug A or active drug B. The factorial is looking at all groups. So neither treatment A or neither treatment B could be a placebo. Then treatment A is, another group is treatment A only, another group gets treatment B only, and another group gets treatment A and B. If you're familiar with clinical trials, this is kind of like, you may have heard the term double blind, double dummy. The double dummy is like, we're going to give them neither treatment A or treatment B. You're going to give them 2 placebos. 1 pill is placebo A and 1 pill is placebo B. Treatment A, they get active A and a placebo that doesn't look right. So they're trying to blind them and double dummy it. I don't know how much more time I have left. Should I stop here or should I keep going?
Alan, we have time till 12:30, so we want to save at least 15 minutes for some discussions. So,
I'm almost done. So selections of groups. So we have active intervention group, and then what's your comparison group? Your comparison group can be placebo, no active intervention. It could be standard or usual care, or it could be an active control, which is an already effective intervention. So when do you want to use a placebo group? We'll use a placebo when you have no standard intervention, or you do have a standard intervention, but the risk of not providing it is minimal. And an issue with using placebo is ethics. Is it really ethical to use it in all situations? So what are some standard of care and active control comparison group issues? Well, you can have many candidate interventions. Which one do you actually pick to be your active control? Sometimes strong evidence rarely exists to guide selection of which intervention. What is the dose or intensity are uncertain. So for example, I have a new drug I want to test, and I want to compare it to, there's already something that's known, like an active control we can use. But what dose is it? Should I use 5 milligrams or 10 milligrams? Like what is the dose we should use that would be good against our new therapy? Efficacy of standard control is often not tested, and their variations are common across, different providers can use different standards of care between its experts and providers, and you can have trends over time. And I mentioned this briefly, but then we have, randomization. What's it help do? RCT is, again, the strongest evaluating effects of an intervention, especially in causality trials. Advantages of randomization: avoids selection bias, and that occurs if the allocation process is predictable. Produces comparable groups in terms of both known and unknown prognostic factors, so it minimizes accidental bias or confounding. And there's two main branches, which we don't have time to get to today. There's fixed allocation, adaptive allocation for randomization. How does blinding help your trial, or masking? So masking of study treatments, so the assignment is unknown to various parties involved in patient management and data collection during the trial. And the goal is to prevent personal bias from influencing study outcomes. And you may hear these different terms. So single blind studies where only the participants blinded. A double blind studies where the participant and the clinical personnel are blinded. And a triple blind study is one where participant, clinical personnel, and the statistician, their data interpreters, as well as blinded, meaning that when a statistician analyzes the data in the triple blind study, they don't know they're blinded. It's like, oh, here's group A and group B. They do not know that group A is placebo and group B is active control. It's only unblinded after all the analyses are completed. And actually, I'm going to skip this last part because I think we're short on time. So I'll just do one or two more slides. So generalizability. How do you generalize? You want to do it to the eligible population. So those subjects who meet eligibility criteria. So you need to consider things like loss to follow-up, reasons for early withdrawal, potential impact of the study results. Consider compliance with treatment regimen and routine clinical practice. And consider how subjects may differ from general and eligible populations. So you want to generalize to the source population. So consider inclusion and exclusion criteria, and consider the screen to enrolled ratio if available. So you don't want to extrapolate beyond your source population, because that's very risky. And then last, consideration for generalization is treatment efficacy, side effects and adverse events, compliance issues, and ability to monitor and assess progress in routine clinical setting. And last, here's some good references, like study design, if people want to look through this and see if that helps them get started on some basic study design. And with that, I think we're free to start taking some questions.
Thank you both. Our next session or seminar is on November 11th, is on Monday, 2 o'clock. These seminars will be posted on YouTube. I'm sharing the link of Penn State CTSI. Of course, our chat or discussion won't be shared on CTSI. So feel free to unmute yourself and ask any questions. We have 15 minutes. Thank you. Or you can post your questions in the chat window also. Please feel free to, Tyler, they can unmute or share the questions in the chat, right?
Yep, everybody should be able to unmute or unmute themselves if they have any questions, or put them directly in the chat.
Yes, very quiet. So yeah, please feel free. We do have 15 more minutes because there's a question. Dr. Honavar, you want to take it? Oh, sorry, you are muted, actually.
Yeah, so I think there are sort of different levels at which you can add AI or ML to your research if it is appropriate. So I think I would say sometimes, maybe you don't really need it. There may be other techniques that might be a lot less onersome to use, but if it is appropriate, then you can do that. So if it is biomedical research, then I think approaching CTSI informatics core would be a good way to get started because we do have staff scientists that might be able to help, assess the feasibility and maybe even generate some preliminary data and so on. Did that answer your question?
Yes, and Susan, I have shared the link to our request form, so you can submit your request and then we can schedule a meeting. There's one more message. Thank you. So any other questions? So maybe we can wait for a couple of more seconds. Looks like everybody is very quiet here. Maybe it is the lunchtime. And again, we will be sharing these on YouTube so you can watch them again. And after the next session, which is on Monday, we are going to have multiple sessions, but they'll start sometime in February. So keep an eye on CTSI newsletter or on CTSI website to get those dates. I think that's all, Dr. Honavar. Alan, thank you so much. So talk to you on Monday.
Thank you.
Bye-bye.
Thank you, everybody.
Thank you, everybody.